One can see the screen. What we'll do today is we'll give you an overview of the program. I will pass this on to Sparsh momentarily, but to give you an introduction, Bioinformatics for Infectious Diseases is a program designed to introduce you to the ways that bioinformatics is used in the study for infectious diseases coming from different types of pathogens. And what we'll do is we'll introduce you to curated examples of how you can analyze data, where to find such data, and how to make conclusions. Okay, so what I'll do in my part of the presentation is I will review some of the introductory material about the program. Then I will pass it on to Dr. Raghavendran, who is going to review some of the practical things of how you can get started with some of the types of data that is usually used for these kinds of studies. So Bioinformatics for Infectious Diseases is a program that is designed for biologists, clinicians, students that are interested in virology, microbiology, and immunology that would like to learn how to apply bioinformatics to different types of big data for research and applications about infectious diseases, to design novel diagnostic approaches, to understand how antiviral and antimicrobial drugs are being designed, how vaccines are relying on data and bioinformatics to address new and maybe potentially in the future upcoming disease outbreaks, and also how to understand what are some of the important challenges and questions that remain to be answered, and how bioinformatics can help us address those as well. So what I'll do is I'll first explain how some of the questions about infectious diseases are related to bioinformatics. You know, if we can think about this program as just a series of questions that one can ask and use bioinformatics to find potential answers. One question might be, what is a pathogen? What differentiates pathogens from non-pathogens? What differentiates, for example, bacteria that coexist together with humans, plants, and other hosts from those that cause disease? How can that help us understand the evolution or the conditions of competition that cause these microorganisms to evolve, share recipes, kind of record what works and how to adapt the tools that they have to overcome different types of challenges? Another question can be, where do diseases come from? How come is it that some diseases persist and some diseases have these outbreaks that are acute, that have a beginning, that have an end? Where do they come from? How do we understand the origins of diseases like coronavirus or Ebola or other diseases that cause these endemic or pandemic outbreaks? How can we understand this in the context of interaction between different hosts, for example, different species, humans, their pets, the different cattle that we grow? How can that be a source of novel pathogens? What is the actual disease that they cause, right? How can we describe the reaction of the host to the pathogen as a condition that we can diagnose? So what makes that as a disease? And we can think of all of these microorganisms as essentially parasites that leverage some of the resources that the hosts have accumulated, leverage their mechanisms of production of vital proteins to be able to replicate themselves, and how they, as a result of hijacking some of these resources, can also cause toxicity and disruption of the host function. We'll also talk about how diseases spread, because what characterizes an infectious disease or a communicable disease and sets it apart from other types of diseases, which are commonly known as chronic diseases like cancer, for example, or hypertension, is the fact that they're caused by a pathogen, and that pathogen is a transmittable pathogen. You can actually share it between individuals. And so when we think of that transmission and the pattern of transmission, we can also use bioinformatics to understand the population scale. How does the population react to a disease, for example, herd immunity, or how does the population become susceptible, for example, having similar characteristics within a particular group or a population, how can that make them more susceptible to particular types of diseases? And we'll also talk about the balance of replication, how while a pathogen causes acute disease oftentimes, one of its objectives is to continue and replicate and find a balance where it can continue to replicate and spread between many individuals, causing them to potentially have mild condition that does not prevent it from continuing to replicate. Then we'll talk about how to treat or prevent disease, and we'll talk about how novel advances are examples of bioinformatics in part because understanding the particular situations where vaccines can be more adapted to the specifics of the pathogen, and maybe novel antivirals could be designed based on our understanding of the interaction between the host and the pathogen, can lead to novel ways to treat disease. But we'll also talk about how early diagnostics, or understanding specifically what pathogen and what strain of a pathogen, or maybe a variant of a pathogen, is causing a disease, is critical for the future of these kinds of interventions. Because as we will see in the last portion, what remains to be done, we'll see how the same principles of evolution can help us understand how the diseases that are caused by pathogens are only preventable if we can develop systems of early detection and understanding of the evolution that they undergo, because we can identify a precise variant and we can treat that precise variant, but we don't always know how to anticipate novel variants. And to anticipate novel variants and design therapeutics that can potentially co-evolve with these pathogens, there are new strategies and novel understanding about how these types of discoveries can really help us anticipate and prevent future outbreaks. So in the context of this particular online program, what we have prepared on our side are a number of examples, or let's call them case studies. Case studies where we can take curated data sets and start designing bioinformatics projects where we have a question, we have a data set, and then we have a path to answer that question from that data set, or methods of analysis of data that could inform some of our understanding of the answers to these questions. So what are some of these case studies and data sets? One of them is going to be about the Ebola outbreak in 2014 to 2016. Now, what's interesting about this outbreak is that it is one of the first examples where large-scale genomic sequencing was employed almost in real time to track an epidemic, to understand how the epidemic is spreading in a particular region, and then use this understanding to design a vaccine and an antiviral drug. Another example that we're going to look at is going to speak about the spread of COVID-19. And when we think about COVID-19, we obviously hear so much about it in the news, but what is exceptionally, I think, kind of for bioinformaticians is interesting in this pandemic specifically, is that the amount of data and the detail of data is now very rich and very precise, being updated almost in real time from many different regions. So really the scale of the data collection here is unheard of in any other kind of disease. Not only that, this particular pandemic has popularized some of the terms that we rely on as bioinformaticians to understand how genomes of particular pathogens or how molecular biology and kind of the background of how these pathogens cause disease are really becoming understood by even the general public. So today, Everyone understands what a variant or a mutation is. Everyone appreciates the ability to study some genomic pattern and try to make inference that has a practical application. We don't have to explain the need to understand and maybe sequence some of these pathogens, because now everyone is in tune with this scientific endeavor to try to really understand how this pathogen is evolving, right? We don't need to explain that it is evolving, right? Everybody knows that. So really, we're going to take a look at some of those data sets in this as well. Another example that we will look at is the development of vaccines to address the kind of chronic or endemic Plasmodium falciparum spread in Africa and in South Asia. We'll take a look at how genomic surveillance and understanding of both resistance to some of the known drugs that are used to treat Plasmodium, as well as the efforts to design an effective vaccine that turns out to be a non-trivial effort that requires a deep understanding of genomic variability across different regions and the understanding of how that genomic variability can be used or leveraged to really find an effective way to interfere. We'll also look at how viruses are not only the result of these kinds of acute outbreaks of infectious diseases, but we'll also see how there's a greater appreciation of the role that viruses play in other types of diseases. For example, in neurodegenerative diseases like multiple sclerosis, where maybe an exciting idea resulting from this observation is that if we treat the virus, if we find a way to treat the virus, the viral infection, we don't have to deal with the symptoms, with the consequences that are the outcome of this persistent infection leading to these long-term deterioration of the cognitive function. We'll also take a look at how these types of observations are relevant in diseases like cancer, where Epstein-Barr virus and others have been found to play a major role in the onset and maybe development of these cancers. So I think really infectious diseases now is going from some of these prominent case studies that we're going to take a look at, like Ebola, malaria, and COVID, and even extend to some of the non-typically considered as infectious diseases, but chronic diseases that potentially have a connection to viral pathogens and others. We'll also look at some of the challenges that still remain to be addressed, even though we think of treatments like antibiotics, for example, as an established way to treat disease that have been very successful over the past several decades. These kinds of interventions are new. Our ability to design vaccines and adapt them to particular pathogens is something that happened in the past maybe decade. Our ability to leverage the antibiotics to treat these bacterial diseases is also just a few decades old. And as a result of widespread use of these types of interventions, we're also seeing the reverse reaction, right? Where we are seeing how these types of interventions can become weaker over time as a result of evolution and adaptation. We see emergence of resistance, and drug-resistant tuberculosis, for example, is a major risk. We can see novel pathogens, bacterial pathogens that are emerging in hospitals that are not possible to treat with antibiotics. We're also seeing novel variants of these viral diseases that also do not respond well to vaccines that were designed. So really, these are some of the challenges that continue to emerge in these kinds of effective ways to leverage bioinformatics. And we'll talk about where we can find some data sets and what are some of the methods of analysis that we can leverage to try and answer some of these questions and challenges. Now, where can you find some of these? We have designed a portal where our team has curated project examples and tutorials that you can leverage as an independent learner, as someone who just wants to obtain some bioinformatics skills, or as someone that would like to apply some of the bioinformatics skills to a particular research question. And so those are some of the things that you will find on the portal, and I will explain today how you can leverage this portal more effectively in the context of this online training portal that is going to provide you with live sessions, as well as some of these online resources that you can leverage to really master some of these skills and practice them on particular examples. So to talk about this, I'll speak about where you can find a particular resource that you might be interested in, how you can find different categories or even collections of different courses, and we'll see how to get started. Now, beyond this, I want to mention that what you will hear in this program is going to be based on tutorials that were prepared in collaboration with a number of experts in this domain. So really what we did is we worked with a number of researchers that are actively investigating all of these topics from the perspective of pure bioinformatics or an algorithmic perspective, how to design methods to analyze such data effectively. We've worked with researchers that specialize in some of these pathogens and have been performing research in the area of malaria or coronaviruses for a long time, and also people that apply the bioinformatics to connect the biological questions with the specific data sets that are available and are being extended and populated with these new viral and bacterial genomes, or even parasite genomes in the case of malaria, to try and answer these questions comprehensively. So we'll leverage their expertise and we'll present to you some of the findings and questions and solutions that they have identified. So what is this program all about? This program, Bioinformatics for Infectious Diseases, is going to help you understand pathogens and the diseases that they cause, where they come from, what are the processes that cause the different types of changes in their genomes that we can observe, how can they cause infection, and how can that infection cause disease. We'll also talk about the specific ways to analyze such data. A lot of this data is going to be focused on the understanding of genomic variability and how to identify specific genomic variants and characterize them in terms of their impact on the pathogen itself, on the interaction between the pathogen and the host, and potentially on the intervention in that interaction. We'll also talk about how you can apply some of these principles to study a variety of different curated data sets, identify the questions that are important for these data sets, and then use the data sets and the methods that we describe to find answers to these important questions. And again, you will have access to some resources that will make your life easier if you're just getting started, but also for those of you that are already more advanced, we will help you connect the dots and really apply these methods in an effective way. Now, the program is going to help you by leveraging two primary resources that I want to introduce briefly before we begin. The first of them is going to be our big data analysis platform. Some of these data sets are going to be exceptionally large. We're going to leverage hundreds, if not thousands, of genomes at a specific time. We're going to look at complex data like RNA sequencing, even single-cell RNA sequencing. And so to facilitate some of the educational aspects of our interaction, we're going to rely on this platform that I'm going to demonstrate in just a few moments. The other part is, of course, our OmicsLogic portal that I already briefly mentioned, but I want to highlight the significance of this online resource because it provides a collection of elements that are going to be useful as you start in this particular program and continue after that, so that you can understand the high-level overview in a short program like we have today, but then continue by leveraging some of the specific technical tutorials to really expand and practice and master these skills so that you are independent and confident in the way that you apply them. So before we begin, what I would like everyone to do is to log in to this portal. So let's go to learn.omicslogic.com. This is a portal where you can register for free. So anyone, even if you're not going to participate in this particular program, you can register. And I'm putting this link here so that all of you who have not registered as of now can take a moment to register. Now I'm going to demonstrate, and please follow along if you have not registered already. So I'm going to log out. The first thing that you will see right here in the menu is this login screen. Okay, so if you press on the login, you can either log in with your email and password. You can create an account. Again, this is a free account. You put in your name, email, and think of a password. Or you can also use different types of social media plugins. So after you have created that, I'm just going to use my own password to log in. And the first thing that you will see if you are registering for the first time is actually the pricing page. But if you go from the pricing page, you should see right here on the very top your email address. And so that's what we want to do, is we want to click on this email address right here. So this email address is your profile. Okay, so please let me know once you have your profile in front of you. I'm going to walk you through some of the things that you're going to be seeing. So just let me know in the chat by saying yes, putting number 1 or something in the chat so that I know that you're with me on your profile. All right, excellent. Thank you, Brian. Eileen. Okay, I'm going to give another 30 seconds for those of you that are still creating your profile. Please let me know once you're done. All right, thank you, Priyanka. Sarah. Excellent. Okay, Anna. All right. So this profile is really designed to kind of connect all of the different resources. Naveen, the creation of the profile itself is free. So really, shouldn't be any worry about cost right now, right? The profile is free. You'll have access to a lot of free resources. If you have questions about pricing later on, please reach out to our marketing team. But right now, I'm just showing you a free resource that you can already leverage from day one. So in this profile, you'll see how the different resources are connected to your learning journey. And we're kind of emphasizing this section because really this is where you can track your progress. You can go back to where you were to pick up where you started. You can always find your certification here, etc. So all of this material is going to be here. What is the expectation of this particular program? This particular program will help you identify the specific topics that you need to master, try to apply them to different projects, and then use the annotation of our tracking of your progress to earn a certificate. And so this certificate is going to be the validation of your kind of understanding of bioinformatics in infectious diseases, number 1. But number 2, it will also provide you with an opportunity to develop an independent research project. So one of our primary objectives in these kinds of training programs is to support research that is coming from a bioinformatics perspective. How to apply bioinformatics to research, that is what we're really going to address in this training program. Now, this training program will have a dedicated program page. On that page, you will find all of the sessions that will be recorded. You will also find a schedule of how to go to the right kind of a topic at the right time, and how to connect to the live sessions that we have today, like the ones that we Now, beyond that, you will also find all of the resources. Now, we understand that many of you are coming from different backgrounds. You might be already an expert in bioinformatics, and you would like to understand how specifically bioinformatics applies to infectious diseases. Or you might be a complete novice, and you really don't know a lot of the topics that we are discussing. Maybe you're not familiar with the terminology, for example. So that's why we include in this particular program also the introductory coursework, like Bytes and Molecules, Introduction to Bioinformatics, and others, where you can start with the very basics and develop your understanding of the different terms that you will find useful as we go through the different case studies. As I mentioned, we will have some live sessions like today, but we will also have recorded sessions. And so you will find those in the schedule, and you will finally be able to interact with our community by asking them questions about the specific topics that we are discussing and getting maybe technical answers to overcome your challenges. So if you ever get stuck, and if you ever have maybe a frustration like always happens in these online courses, I don't understand, I can't do what I'm being told to do, you can always reach out to us. So our team is going to be there to support your journey, and you're going to be able to then go through the specific tutorials that we recommend to learn about that. Now, if you don't see any courses, that is because you are likely just signing up. So if you go to this page, the course tab has the courses that you've already started, okay? If you want to find a course, you have to go to the top menu, and this is our library of resources, and find the relevant course. For example, if I would like to learn about COVID, I will find SARS-CoV-2 genomics as an introductory project right here. So once you start this, and you start by clicking on the lesson, and now you've started this project, you can learn through and then click on next, and eventually that will appear on your profile as well. So Christine, hopefully that answers your question right there. Okay, so finally, before we go on, I want to recommend you to connect to our community as well. So what this program represents is a community effort. Not only are we going to have these kinds of sessions and interact with you, try to help you, try to explain some of these topics in an accessible, approachable way, and really distill these complex topics to some simple exercises that anyone can perform. We're also there to connect a community of learners to opportunities. And those opportunities might be professional opportunities to develop your career in bioinformatics, also opportunities to connect with potential research collaborators. So we're going to introduce you to some of those groups. And today I'm just going to share here in the chat 2 of our groups that you can connect to. One of them is on LinkedIn, and another one is on Facebook. For those of you that are on either one of those platforms, we welcome you to connect with us personally, but also to connect with the broader community that we serve. We have over, I think, 1,500 people on LinkedIn, over 10,000 people on Facebook. So you can really leverage this community to not only learn about bioinformatics for infectious diseases, but then also to continue and apply bioinformatics to other domains and to advance your career or find opportunities for research. So today's session is going to be dedicated to next generation sequencing. One of the technologies that has really advanced our understanding and ability to treat and diagnose diseases like infectious diseases is our ability to sequence genomes and transcriptomes and understand what's going on at the molecular level. So when we think about next generation sequencing and the way it has become popular and maybe widely used as a technique, this has happened in the course of the past couple of decades. And today, with decreasing costs of sequencing, it is being used in remote areas where some of these diseases are originating, where some of these diseases are being changed and transformed, but also in most kind of technologically advanced places, it is becoming a more and more reliable and useful technology to understand infectious diseases. So to learn more about this technique, I'm going to invite Dr. Raghav to speak about how next generation sequencing works, what it contains, what kind of information it contains, and how you can potentially start learning to use it, to actually apply it in the context of infectious diseases. So Raghav?
Yeah. Hello, everybody. Let me find ways to share my screen.